So we're very happy to have Angela Reynolds here to talk to us about her work in modeling the immune system. So Angela got her PhD from Pitt, University of Pittsburgh in 2008 under the supervision of Bart Armentrout. So after that, she got a nice tenure track position at VCU, and she's rose through the rank and is now the director of the PhD program in system modeling and analysis there at VCU. So I was just looking at one of her very recent work on COVID, where she talked about the role of macrophages in lung inflammation through modeling. So I thought that's very interesting. So what she's going to talk to us about is modeling the innate immune cells. Thank you, Angela.
Thank you, and thanks for inviting me and spending some of your Friday afternoon with me. So I'm going to kind of go through a sequence of models that we built where we kept increasing the complexity in the innate immune system depending on the disease, and really trying to tie into data that was available or we think is going to be available with some experimentalists that we had collaborated with. So so to motivate what we were doing, we look at the immune response because it is a system that we need it to respond. You know, we see right now with COVID how their response can vary from one individual to the next, but we need our immune response to respond to be able to fight these external insults, whether it's a pathogen or bacteria. Right? But we needed to do it in such a way that there's a delicate balance between what's upregulating and taking care of and removing that insult at the same time that we're building a anti-inflammatory response to regulate that. And a lot of this, you know, the immune response evolved when we didn't have antibiotics and these kind of things around. And so our system is very primed to react to these bacteria and viruses. And so that anti-inflammatory has to be able to regulate that. And when you have a mismatch there, or you don't transition into the anti-phase, or it's not strong enough to regulate, or you don't mount enough of the pro stage to actually fight the insult, you can end up with outcomes that are non-ideal, right? And so we wanted to be able to really capture components of that immune response, and we kind of are in building what we think is a module that is very adaptable to various diseases. And almost all diseases, if something is going wrong, your body is going to trigger an immune response to take care of it. And sometimes it even triggers the immune response when nothing, right? Nothing is going wrong. It might attack your own thyroid, it might attack, you know, respond and you have allergies or something like this. And there's, you know, two components of the immune response. The innate is the non-specific. It's that initial response that your body isn't fighting in a way that is specific to the insult. It's the generic response that starts up and then recruits this secondary adaptive or specific response that gives rise to cells that are really good in fighting that particular insult. And that's what we rely a lot with, you know, with vaccines and whatnot, to avoid getting really sick. We build that adaptive response without ever actually having triggered the, I mean, we may trigger the generic response, but we have the adaptive response ready to go. And so there are several T cells that are a part of this immune response, right? So there's key cells, neutrophils, macrophages are one of the big ones, or the big two in innate immune cells, so that early responders. And then the T cells are a big part of that adaptive response. I just want to kind of give our idea of how these things come together. So macrophages are what we call an APC, which is over here on the left, and that's an antigen presenting cell. So what they do is they eat up the invader and present components to the adaptive response, the T cells, in such a mechanism that they, you know, a lot of times we refer to like these enzyme matches and lock and key type scenarios where they're going to match to a particular T cell that knows how to fight that particular type of virus. So the antigen is generated from breaking down the virus or bacteria. It's then presented to the adaptive immune response. It triggers those T cells, which are good at fighting that particular insult. And it gives rise to many different components in the T cell, there's different types of them. Some of them are more just ready to fight, and some of them are more regulatory and building that memory so that the next time we see this insult, we're able to fight it. So being able to elicit a innate immune response and trigger this, and then have this all well regulated and not lead to a strong inflammatory response that keeps you with an elevated level of inflammation and is harmful. And I think this is a little bit of, many cases of what you're seeing with COVID. They're not worried about the fact that the virus is still alive and well. They're more worried about the fact this is all overactivated and causing damage to organs. And so a lot of the therapies, I think, that are being used or tested, initially, and I should have checked before I spoke today, I think they're still being used, are attacking this IL-6. I just saw, you say this IL-6, I just saw a paper that was called Cleanup on IL-6. And it was related to regulating this particular signaling protein or cytokine here that is a part of this adaptive response. So understanding a patient's outcome during disease often requires understanding the immune response and kind of where that individual's immune response was, right? You can imagine if you're already sick and you get sick again, right? If you're already elevated immune system, or you're coming off of a disease and you have a high levels of anti-inflammation, so it's harder to mount another response. All right, and so what we've done is kind of started by building from a reduced model that had lumped immune cells into more and more specific cell types, and coupling this with parameter estimation and sampling to kind of look at how what are viable dynamics that come out for different parameter sets and how these affect the actual overall outcome. Right, and so, sorry, I keep clicking and it's not actually advancing. This was my initial project, and I'm not going to spend much time in it, but I just want to give this kind of overview of when you lump everything together, what are the main things you're really thinking of? You're thinking of some kind of initiating event. We called it pathogen. It's a bacteria or virus. It causes inflammation. I still regret the choice of this is supposed to look like fire, and a lot of people don't see that. So it's inflamed. It's your immune cells are coming in. If you think of a wound, that red area you get around your wound, right, is really because the immune cells are influxing in, and it's causing associated tissue damage. And so you have this damage of those cells that then causes for more inflammation, plus the inflammatory response actually upregulates. It may trigger an adaptive immune response. Regardless, right, you've got this feedback through itself and through this other variable damage, but these are also contributing to the recruitment of a anti-inflammatory response. And so these are kind of these two different responses are competing with each other, and you would hope that things would resolve in such a way that the pathogen is removed and the inflammation goes back down. So we developed a system of 4 ODEs. Few nonlinear terms here. The blue is showing what was actually inhibited by the anti-inflammatories. And, we had growth dynamics on our pathogen. We thought of a background localized. You can think about your antibodies in your skin or something like this that can handle eliminating small levels of pathogen. So really low levels of pathogen, even in the absence of recruitment of this N star variable, will be cleared by the local system. But any large amounts of pathogen will elicit this response or will need this response, let's say, to actually be able to regulate. So then what we did is this is a bifurcation diagram looking at the growth rate of bacteria, and we spent a lot of time trying to make sure that we actually had this tri-stable region, because this is what you would possibly, expect to see clinically. And so the idea is for low growing pathogen, your body's always going to be able to eliminate the insult, but you may end up over triggering your immune system. So this is what aseptic death is. This is a high level of inflammation, but without the pathogen present. So low growing pathogen aren't going to get out of control, but if you give, and this is obviously going to be dependent on initial conditions, right? You can end up in a scenario where you triggered so much inflammation that the positive feedback loop is keeping you sustained. Higher growing bacteria, you can end up in a scenario where you can get all three. And this third case here is septic death, is where your bacteria is actually still present. And so the immune system can't control it. It's still present. The immune system is upregulated, and we call that septic death. A lot of times you don't see this clinically because you would intervene with an antibiotic or something like that. But a lot of this work came with working with people that were working in liver transplant ICUs at Pitt, and they were seeing lots of cases where it would lead eventually to multi-organ failure, but where you might control the initial result, but you ended up in this case where you had a sustained inflammatory response. And so this model formed our kind of thought process to start to address specific diseases. So we looked at first expanding out that inflammation into both the macrophages and the neutrophils and focusing on wound healing. So we'll spend a little time with that. Then we moved into wanting to really understand this sequential response of these key innate immune cells. And so this was peritonitis, and this procedure that we did experimentally here is actually what they do when they want to collect macrophages for other experiments. But here we collected at multiple time points and looked at what M1 and M2 were, and I'll get into what those cell types are, and fit this kind of, stereotypical sequential response of immune cells. And then we also took that similar construct and moved it into lung inflammation and the blood. And here we're focused on mechanical ventilation, and this is where we're really starting to, and I'm not going to, future work type thing is to kind of build in this mechanical ventilation in such a way that it's age dependent and hopefully link it to some of the stuff we're seeing in COVID. So just a shout out to my graduate students. We're a very new PhD program, and Rachel was my first student, and then Marcella joined the team, and now Sarah is finishing up her project. So the immune system module has grown as the years have continued, and we've kind of handed it off from one group to the next, and everybody gets to kind of add their cell type. So on that wound healing project, that was when we first broke out the innate immune cells into the neutrophils and macrophages. And we used that to look at what happened when you elevated cortisol, a stress hormone. And that was of interest to our collaborators because of a lot of wounds may come under stressful scenarios, especially scenarios where you can think of a soldier or trauma, right? Any kind of trauma, your body is going through a stressful event that led to the injury, so you have high levels of stress hormones as you're trying to heal. And macrophages in particular are very sensitive to cortisol. Also, we found this out, I never realized, but we found out that immune cells actually have receptors for estrogen. And so depending on whether or not estrogen is present, they behave differently. And so if you want to be able to model what's going on in a particular individual and their immune cells are going to change depending on whether or not there's estrogen present, we needed to include that in the model. And, we actually, it took a lot of a more thought process than you would think in trying to work with the data, because turns out that they do all the experiments on female mice. Now the NIH in particular has rules where you have to do both genders. But back in the stuff that we were looking at, it was all with estrogen because females would heal faster because the estrogen would give rise to a less influx and therefore less collateral damage of these immune cells. And so they would heal faster. And so it was easier to do experiments with the female mice because they would, they would heal faster. You wouldn't have to run your experiments as long. So that's kind of how we stumbled upon this and thought, well, we should probably include that. So I'm not going to go into all of the equations and derivation for both of those, but what we were really focused on is these first two stages of this. When you actually get the wound, your blood releases a lot of key factors in that initial event, right? You get a cut. Pretty much any cut is going to have some bacteria in it unless you're having a surgery, all right? And so you're going to have some bacteria along with the tissue that's actually damaged and the components of the blood that are released at the immune site. And then you're going to elicit an immune response. And you see in this diagram here, the neutrophils come in first and then the macrophages, and these later stages are more associated with the repair and the remodeling, and we focused on that inflammatory stage. And so we took very similar equations to what you saw before. All right, here again we have that scenario, and I put in the details here a little bit more, right? It's just a logistic growth term which would only have that P infinity being stable, but when we added this localized antibodies, we introduced a bistability, and then we added to it the recruitment of the different immune cells that would help control those larger insults. And we didn't just think the pathogen would be an insult, but we also included here P sub T, and this was associated with those platelets and the debris that would be in the wound. And so both of these are coming together to elicit both the macrophages, so this is the recruitment term on the macrophages, they're responding to the platelets, the bacteria, they're responding to downstream's effect of their own activation. So as they become activated, they're going to release cytokines that we don't explicitly model, but we attribute those to the level of the current pathogen, and also those of the neutrophils. And the neutrophils also respond, but we don't have them self-recruiting in this model or responding to the macrophages, just more that initial response. And again, we have inhibition, and we're associating this inhibition we see here actually with the neutrophils. So when neutrophils are present in a wound or influx, the way they attack the foreign insult or help remove the debris and damaged tissue can be very toxic to the local environment, and it actually makes it quite hard for the macrophages to do what they need to do as antigen presenting cells if the neutrophils are elevated. And so this inhibition is associated with, those high levels of neutrophils kind of down regulating what the macrophages are capable of doing. And so here we went a route using Latin hypercube sampling. We selected sets, so we sampled over, thousands and thousands of sets, and then we accepted sets that actually had healthy as a stable fixed point. So we had derived, doing a stability analysis, what the conditions on these equations were for a stable, healthy fixed point, and we thought that, that should make sense, right? Your wound should be able to heal, and if it heals, it should stay there. And additionally, we took into account some constraints that I'll throw up on, I think they're on a later slide, but, and these were mostly things that we got from the literature that related to source and decay routes, and they were kind of built into the method of our sampling. We would select one variable and then, select one parameter, and then within that same set, we would make sure that when we selected the next, it satisfied whatever condition we had related to the source and decays of these neutrophils and macrophages. They know things about macrophages and neutrophils such that the activated ones, how they behave differently towards the rest versus the resting ones. So like this is the source of resting cells, and this is the decay of resting cells. for neutrophils, but we also had the decay of activated cells, and we made sure that those lined up correctly. All right. We also did selection based on the resulting dynamics, not just the parameters, but when you actually simulated the associated results, we knew a lot of the data, the way these experiments are done, different mice have to be sacrificed at each time point. And because these experiments kind of take a long time, because it takes almost 2 weeks usually or 10 to 14 days for a wound to completely heal, there's a lot of data at day 1 and 3. And so a lot of this stuff, data was saying that neutrophils peaked at day 1, but there really wasn't much experimentally anywhere until day 3. And so what we did is and there was some early on data. So we said, yes, the day 1 data was higher than what we saw at day 3 for neutrophils, but we don't really know where the peak is between those 2 data points. And so we accepted that the peak should be in the range, and we kind of used the data that we had to determine these ranges, or data that not directly from our collaborator, but from the literature. And the same on the macrophages. They tended to do those experiments in such a way that there was samples from day 3 and day 7, and day 1, and they knew the macrophages were highest around day 3. And so we put our window such that it was kind of between those data points. We also were looking for the differences that would arise in these macrophages being resolved if they're going to resolve out to day 20. And so we selected for that, and there we get here just some of the sample transients that came out of this model. And what we saw in the parameter structures, which wasn't structure we saw in the parameter sets that were accepted, which probably wasn't too surprising because of some of the conditions that we were enforcing, right? These were the most correlated parameters within the model. And this is the source of resting cells and the inhibition coefficient on the neutrophils. All right, so how effective neutrophils are at inhibiting macrophages. And then here, this is, again, the same source, but here, this is the recruitment of neutrophils by platelets. And you can see their selection process was, related. And so none of this was super shocking that they were, somewhat correlated. I think that this structure worked out so nicely was a little bit surprising, but what we did see is when we did this selection, and I showed you what we picked for our selection criteria, what we did find was that the neutrophils were pretty much very close to day 1 in many cases. The minimum for the sets was peaking around 3/4 of a day and out to 1.69. All right, but the macrophages were kind of a little bit a day later, and really there was the whole entire range that we kind of selected for was still present. But if you go back and look, the neutrophils, we allowed them to go out to almost day 3, but the sets that came as being acceptable dynamics overall didn't actually go out to that window, right? There were no acceptable sets that were well behaved with regards to the macrophages and stability of health that actually gave rise to neutrophils at that tail end of the window that we selected. And this was kind of a, very, I would say, preliminary type of validation. We weren't actually kind of fitting the model to particular data, but what we did was we took a representative set. So these are one of the set within the collection that was accepted that was closest to the average behavior of everything else. The actual average did not lie in the set of acceptable ones. I don't think it had stable health. If you took the average over the parameters, it didn't actually have dynamics that gave rise to something that would actually be in the set. But we found the set within our acceptable sets that behave most like the average behavior, and kind of use that just to see, does it do what we think it will do in relation to blocking either macrophages or neutrophils. So in this experiment, they blocked macrophages, and what they saw was the neutrophils get very similar peaks, but their tail is extended. And if you see that, you know, our peak is a little bit lower, but still very similar time. So it's lining up with what's going on here. And then this tail is being extended, just like the tail over here is being extended. And then on this bottom part, we have the no neutrophils and with neutrophils. And if you look, this is only data from one day early on. There wasn't any time series data, and it said that at 24 hours they should still look the same, and we were able to capture that, but we didn't have much for any later time points. So again, we built off of this representative set the effects of estrogen and cortisol, and then we sampled over the cortisol and estrogen parameters, and for the sake of time, I'm not going to get into all of this. But what we saw there when we looked at the acceptable sets, and here we accepted for known dynamics in as far as changes in macrophages and neutrophils when mice were restrained. So they made it so they couldn't walk around, which caused them more stress, and that affected their ability to heal. And here, the way we did the sampling, CM2 was the control. Well, let me say first, CM2 and all of these C's control the effectiveness at cortisol inhibiting different things. So this is the inhibition of the neutrophils. This is the inhibition of macrophages. And let me just go back, right, on CM2, that's the ability for macrophages to be activated, versus CM1 is the ability for macrophages to phagocytize or eat up the debris and the pathogen. And so the way we selected, we knew that one of these was more effective, so it's not surprising that the CM2 causes more of a change. I think so if you look at a fixed CM1, if you follow this up, right, there's a lot of change in the color. The model's very responsive to changes in CM2. What I found that even despite whatever range we chose down here, it really had almost no effect. That was kind of more the surprising part that we came out of this, where changing that CM2 parameter did not change much at all to how much the macrophage population was reduced at its peak. This is looking at the change in the total peak as kind of a measurement of that total response. And again, we did the same thing up here. We're looking at the percent decrease in the peak for neutrophils for various levels of this inhibition. And so, you know, the lower this is, the more effective the inhibition is. And so these lower numbers give rise to really high percent decreases. So again, just kind of giving, I would say, this first step to separate out macrophages from neutrophils and kind of getting a feel for how they work and the parameters that are associated to them, and thinking about, in some cases, the parameters and sometimes thinking of the resulting dynamics. And then we took that concept and we moved it over to this peritonitis. And again, this was more focused on looking at expanding out the macrophage population. So macrophages actually can be activated for different types of behavior, M1 versus M2, and it's really thought of being a spectrum. And so you can, you know, think of something that was purely activated to be M1 is going to produce a lot of pro-inflammatories. They're going to be destroying the pathogen. They promote inflammation. If you were fully activated to M2, you would be dampening and you would be producing anti-inflammatories. Things can get signals from both and kind of live in the middle. And the only thing that they both do for sure is phagocytosis. So you've got this two different phenotypes that are in some sense competing with each other, and you can transition between them. And so what we did was we wanted to understand, in a very basic bacteria giving rise to an immune cell response, you know, can we fit that system? So here we're very focused on actually fitting particular parameters to our data. We worked with someone in internal medicine. She does stuff with macrophages, using different genotypes of mice with different genotypes and their macrophages in relation to atherosclerosis. And so she quite often takes macrophages from mice. So how they do this, right, is they give a broth solution into the peritonitis, which they're injecting broth nutrients into the gut, which has bacteria that elicits a growth of that bacteria that's above the normal levels, and therefore elicits an immune response. And so what we did was, in hours here, we collected samples from mice. Oh, my numbers look like they slightly shifted over here. And had some key numbers where we thought with regard to neutrophils, key numbers with regard to macrophages, but kind of covering that whole response. And at each of these times, she took the samples and did flow cytometry and determined the portion of the population that was M1 versus M2 and neutrophils of those immune cells. So this is a lot more complex system when you start introducing M1, which in some sense behaves a lot more like the neutrophils because it's very pro-inflammatory, and transitioning between M1 and M2. It's thought that the M2 to M1 does not happen very much. We did include it in the model because there wasn't a consensus there biologically. It ends up being not a hugely important parameter when we go through everything. So it's probably not playing a huge role. But we have this broth stimulating the pathogen. The pathogen is recruiting these blue arrows, or the upregulation to these immune cells. And the neutrophils themselves are becoming apoptotic, which is a programmed death, which is more ideal than a cell taking on too much bacteria or something like that and exploding, and then their contents elicit even more of an immune response. So this is a programmed cell death, and once they go into that state, the macrophages actually clear them out so that they don't cause further inflammation. So these components were all incorporated, and this cell was brought in because the more that they actually eat up apoptotic neutrophils, it drives them towards being M2. Because that's, if there's a lot of apoptotic neutrophils going on, that means that they're kind of done their job and they're going through their programmed death. And so we're shifting more over to repair. And so here we have a activation type term. As you see, it comes from assuming quasi-steady state on the resting. That was throughout all the models. All of our activation type terms have this structure. And here this switch from M2 to M1 as a function of the apoptotic neutrophils is here. They can also just switch for, just as a natural process outside of the AN, and then they decay. And here are these main things that are causing the rate of activation are activation by pathogen, the byproducts of the neutrophils, right? I told you they can cause some collateral damage that elicits more of a response. Also, there's production of cytokines by the actual M1s, and then those neutrophils that don't go through that apoptotic and they become necrotic and they spill their cytokines when they die. That also elicits a response. Versus the M2 activation is really just a self-promotion of the M2. And again, the apoptotic neutrophils drive the M1s to the M2s, and then the M2s will kind of further elicit their response. Sorry, wrong way. So what we did with this model is we spent a lot of time fitting and using sensitivity analysis. So we estimated all the parameters using an iterative weighted least squares method that generated a particular fit. For that fit, we went through and generated a sensitivity matrix numerically. We ranked them due to sensitivity. We eliminated those that the system was not sensitive to. We used 5% of the maximum sensitivity to determine the threshold from below which we didn't include. Then we went and looked for collinearity within the resulting parameters. We knew from the sensitivity matrix, since it had rank 6, that we were looking for a set of 6 parameters to form a identifiable parameter set that we could then use for our final fit. And so we chose one group from to be our identifiable subset, and I'll go through these details a little bit more on the next few slides, and then finally went and estimated and just had those final 6 parameters that we were allowing to vary. So the initial run through, we had this full model. Here was the threshold for our sensitivity, below which we didn't include them anymore. Then we focused on these parameters. And here you see we looked at groups of 6 and calculated the collinearity index for each of those groups. We only considered it to be identifiable if its collinearity index was below 20. And so those are the ones you're seeing here. And then from here, we chose actually the one that had what we thought were biologically understandable, or things that you could think of as actually being able to change biologically, or well understood biologically, not just mathematically, to have in our set as a way to kind of think about how would we modulate that response. And so here are the fits. These are our fits on our M1, M2, and N. This panel just shows you the ones that where we didn't have data for, what they looked like, the pathogen broth and apoptotic neutrophils. And you see over here, these were the 6 that we eventually chose. And we did also make sure that the M1, M2 transition parameter was in that set. And then what we did is we went through and looked at, in that final fit, what was the sensitivity for different two parameters for different attributes of our transient. Like what affected the peak amplitude the most. So if you look over here, the peak amplitude of M1 is actually greatly increased, or increased most, right, by modulating the source of the resting neutrophils, which allows more neutrophils to come in, which elicits more of the macrophages. Second to that is its own resting population, so giving rise to more cells that can be activated. But this is maybe slightly non-intuitive, right, that in changing the neutrophil population is actually more going to change the M1 population more than actually changing the resting macrophages. And those resting macrophages are splitting off to M1 and M2, so that's also playing a role there. All right, there's other attributes we looked at as far as M1 total cells. If they're going this way, right, that they're causing a decrease versus an increase. And this SNR is important. And we didn't, I've cut off some of these graphs. They're, you know, in the paper, I think we go through all the parameters, but I just focused on the top here where we actually have some bars that are visible. All right, and so we did this not only in that base case, but we also then looked at what happens in a scenario where this is the case that we fit to, but what if we modulated, so we made it a state where we had a poor outcome, all right? So we stayed elevated instead of resolving. And in that poor outcome, how do these different things, these different attributes, the M1 numbers at the last time point and the M2 numbers, right, attributes of the transient, how do they change with respect to modulating the parameters? But here we've overlaid it for both different outcomes. And here the thought process is, right, if I'm on this red curve and I want to modulate and get myself down here, right, I don't want to pick a parameter and change it that's going to actually cause a huge significant change in that lower outcome, right? I don't want to be modulating something that I'm going to just end up in the middle here, right? Or something like that. I mean, I'm probably not explaining it in full detail, but we kind of want to understand how what we're going to modulate is going to affect both the healthy and unhealthy outcomes that we're trying to get to. So that's what we did in this. And then we chose a parameter to actually modulate and try to save our system. So here's the baseline case where we are not doing well, and then we modulated different parameters to see if we could save the M1s and save the individual. And so here we spent a lot of time in this model really developing the M1, M2, and in this kind of very generic bacteria gives rise to sequential immune cells, did a fit, and we're kind of using that now for the basis of moving it to different diseases. So we're looking at it in atherosclerosis with my collaborator that actually did these macrophage experiments. She's interested in plaque formation because macrophages transition into foam cells, which get sticky and cause plaque formation. And if those plaques rupture, you have heart attacks. So we're working on that. We also look at ventilator-induced lung injury. And so when you go onto ventilator, we probably heard a ton about them after what happened in Italy with so many people needing ventilators at the same time due to COVID, right? You're forcing air into the lungs because something is wrong, right? Something is going wrong, but if you don't do it, you won't be able to get oxygen where you need it. And unfortunately, though, when you're forcing air into the lungs, you're going to cause mechanical stress, cause local inflammation, and that's going to cause cellular dysfunction, right? And we're focusing here on this cellular. How does the stress and strains that's going on in the lung give rise to epithelial damage that recruits an immune system? And long term, right, this can cause things like more multi-organ failure because you're eliciting such a strong inflammatory response that other organs are getting attacked by your immune system. And so I know I have to update these stats because these were pre-COVID, but there are a lot of people that need ventilation. You're pretty sick when you go on there, so the fact that there's a high mortality rate is probably not super surprising. The cost of it is quite high. I mean, it costs a lot to have people on ventilators. They have to be sedated. It's unnatural to breathe when you're on a ventilator, so they put you to sleep. And another key thing there is a lot of people that need ventilation are older. And so we originally started in this project, we had a grant from the National Institute of Aging, and we were looking at how different mice of different age respond differently to ventilation. What I'm going to talk about today doesn't include age yet, but that's our next step, is adding in age, right? Understanding, and a part of that is more your initial conditions. How are your immune system when you first go on the ventilator going to affect in pretty much after I think pretty much after the age of like 18 or 20, your lungs are actually aging. They've stopped developing, and you can think of it as you're kind of developing scar tissue, and they're less willing to, and they're more resistant to that air being pushed in. So the immune response is quite different with age in the aged lung. And so here we took that same structure. We didn't assume a quasi-steady state on these resting cells anymore, but we do have both macrophages that are in M1 and M2. And I should say, the way the experiments are done, it tells you which of the portion of the population that is expressing M1 type markers. So always fitting to the portion of the population, not saying that there's an exact count of M1, because as I talked about before, we don't assume that they are actually either M1 or M2. They're kind of a mixture of them. So we're always fitting to the overall expression of M1 versus M2 phenotype. And this one is the naive unactivated one. And we didn't put that in quasi-steady state because that can be recruited locally. And we wanted to have this compartmental model where we have all of these cell types that can come from the blood and go the other way, diffuse into the blood to elicit a further response, are explicitly modeled. So we added this additional compartment, and then within here, because of the communication between the epithelial cells that are healthy becoming damaged and transitioning to dead in severe cases, are producing pro-inflammatories and mitigating. On the reverse side, the macrophages are signaling that it's time to repair and promoting the division of those epithelial cells to form a healthy tissue. We actually didn't explicitly model particular cytokines, but we included them separated out from the immune cells. So instead of just saying M1 has a positive feedback loop, we say M1 produces pro-inflammatories, and then the pro-inflammatories cause further M1 response. And a lot of this is just to help with the crosstalk between the epithelial and immune cells. So the equations start to get, a little bit more complicated. We have damage to the healthy immune cells by both the M1 and M2 neutrophils. When they influx in, they're going to cause collateral damage. We've had kind of a similar term in every model. Those cells become damaged, and I didn't show the ED equation here, but once they become damaged, they have some baseline repair. The repair gets upregulated when you have more repair mediators. So this is this repair associated with the repair. So if I pulled out the ED on this term, you'd have BR plus some upregulation, some baseline elevated, similar to what you see here. There's a baseline rate at which proliferation is occurring, and then that's upregulated when there's pro-inflammatories. And the idea there is these alveolar type 2 cells are what are going to form the alveolar sections in the lung, and the type 2 cells, all they do is there's actually type 1 and type 2. I always think it should be the other way around, because type 2 divide and then those cells become type 1, and they are a majority of what is in the alveolar. There's a subset of them that is actually able to proliferate, and when there is damage to the type 2s that are nearby, damage to the type 1s that are nearby, these type 2 cells get activated. And so that's why this pro-inflammatory causes this baseline to go up. And contact inhibition, you can only go so high. There's only so much of the space, right? Those type 2s aren't going to keep dividing once we have returned to our full population level. And then here we actually just have a very generic, when you're on the ventilator, we're going to cause damage to those healthy cells. So you have this epithelial interaction, baseline repair, proliferation, and then feeding into it's what's going on with those neutrophils and macrophages. Their main goal is to actually decrease the damaged cells, but they do that in a way that they remove them and it becomes dead space, empty space. Again, you have differentiation to M1, and this is being upregulated by the pro-inflammatory mediators, but inhibited by the anti-inflammatory mediators. And this is diffusion term. The epithelial cells can't move in and out, but the immune cells and mediators can. And again, we have that switch from M1 to M2 due to phagocytosis. So we had what resulted in 3 different types of outcomes. And in this scenario, we are starting with proportion of epithelial cells. So of the total epithelial population, how many of them were healthy? So we're starting pretty high. We're turning on the ventilator, in this case for 2 hours. We're relating this to mouse data, and 2 hours of ventilation for a mouse is actually a pretty long time. And then we're turning it off. And here, this case, we go back to healthy. This case, we elicit such a response, and these are different parameter sets, not different levels of ventilation, but different underlying parameter sets for those epithelial and macrophage dynamics. And here, when you turn on the ventilator, you elicit such a strong immune response that you're down here. All right, versus in this scenario, this one actually starts out pretty inflamed and just returns back to that. But what we did was we kind of classified not only where you start, but where you end up based on the epithelial cells. We also, our data, as I said, was more for that M1, M2, and those naive cells. And we only had initial conditions and at the end of the 2 hours. And we didn't want to, I would say, we're using the data as more of a illustrative example of what you could do with data, but sorry, I just heard a beep. I don't know if that was, I can't see the chat. Anita, can you let me know if that's something I need to address?
What do you want me to, what do you need?
I just heard a beep. Was it through
No,
like chat or something? Okay, sorry.
I didn't
It must
hear
be
anything.
something else on my computer. Sorry about that. And so I'm going to talk a little bit about the data, but we didn't want to fit to just these 2 data points. And as you can see, there's huge standard deviation bars on these, but more just using it as an illustrated example of what you could do with data and the need for more data. So we have our M0s, our M1s, and our M2s, and you can see that associated with these outcomes, we're classifying them based on what's going on in the epithelial, but you get very different dynamics in all the different macrophages too. And so what we did is we did like Latin hypercube sampling again. We wanted to start in a steady state. We were looking for initial conditions. We didn't put any constraints on them. You could think about going on the ventilator sick. You could think about, your lungs being fine. Maybe you had a brain injury and you need ventilation, right? But your lungs are fine. So we didn't do any kind of selection other than the fact that you were in steady state. And again, right, if you're already if you're actually having a disease like COVID, you're probably not in steady state because your inflammation system is changing. So that's something we'll have to address. But we were kind of looking at how do the underlying parameter structures, thinking more in terms of age, you know, affect what happens to the ventilator without even trying to think about, you know, if there was an actual inflammatory response that was progressing, just trying to understand the ventilator induced component here and how your initial state might cause different responses. So here, this is what the ICs were, how many were healthy, persistent, or dead, versus your end state. And we didn't simulate all of them that, you know, if you were below a certain amount of epithelial cells, we didn't even put you on the ventilator. Right, you were really dead. It sounds terrible but again, we're talking about simulations here. And then this just breaks down into who fit we just looked at the M0 data. That was the one that was, I think, probably most consistent with what we thought should be going on and had a lot less standard deviation. So we separated that out versus fitting all initially versus fitting all at both time points, and kind of just went through and looked at how the different outcomes versus where we were initially. What's in parentheses is how many of those sets actually change states. So 415 of these change states. All right. For here, though, only 3 of these came from a different state. So, right, this is the healthy state. For somehow, you know, ventilation actually helped 3 of these people. Right. So that's a little shocking, right? But ventilation hurt 415 of these people because they left the healthy state. So again, what we did was we wanted to really do like we did before and look at the parameters and at the same time understand the resulting transient dynamics and try to figure out if there's something that we can figure out or look at with these, you know, virtual cohort of individuals that might be a good predictor or indicator of what's going on. So initially we just took the parameters, looked at what was correlated, and here you're just seeing some plots of, again, the most correlated ones. Here they're showing the the resolved versus persistent. So these ones have resolved back to health versus persistent inflammation, and you can see how those sets lies. And there was really no selection criteria that forced this curve. And again, you can see there's a bunch of blue ones over here, but this somewhat seems like, you know, a little bit more could be done or looked into of what's going on here. And here you still you have a very similar trend in this orange and green, and for the green ones, it's, you know, a lower. What we're seeing here is at the end of ventilation, what is the ratio of the healthy epithelial cells to where they started? So in some sense, what was the total amount of damage caused by the ventilator? We also just did some, you know, statistical analysis and looked at which of the parameters were significantly different between different groups. So, you know, between healthy, persistent, healthy, dying, all the different combinations, we wanted them to be different. And you can, I'm focusing on these because these kind of came up in all of our types of, you know, methods at which we looked at these. And the baseline repair is a huge factor in how well you do. How quickly you're able to recover even in the absence of the immune response is playing a key role. And this is how that collateral damage is. So how much you respond to those macrophages and neutrophils. And these were very key things in determining your healthy, persistent, and dying. We also did a sensitivity analysis. This we used EFAST, which came out of Denise Krishner's lab. There's 2 different things that are calculated. It uses sampling, and here it's looking at varying each of these parameters independently, and we're looking at the sensitivity of the transient at different time points. Versus here, it's varying other parameters at the same time. And the way I kind of interpret it is this top panel, if you're only going to be able to change one, really kind of gives you intuition on what is going to have the most effect as a treatment, as in modulating it. You're fixing everything else. And this, I see the bottom panel, since more things are varying, which is really kind of driving the patient-to-patient variability that you're seeing within the cohort. And again, the BR comes up as important over here. My Zoom window is making it hard for me to point on the bottom panel here. And BR is very important down here. And, you know, this may give you some, right, if you can elicit some kind of repair that's, you know, baseline. The question is, what happens when you go back and take off the ventilator, right? Do you end up at a weird steady state or whatnot? Is it a drug you need to keep going with or whatnot? So there's a lot to be done in exploring these kind of things. The next thing we did was this used decision trees. It used what's called a random forest by generating several decision trees. I think we used 1,000 for, sorry, I'm blanking right now on the exact number. And so the idea is you're developing a decision as to what is predictive of outcome, right? If you look at KMNN, can you make some prediction of what outcome you're going to go into? And so if these random forest had a collection of decision trees, and while the decision trees were being generated, if this was an important key point on your tree in more and more of the randomly generated decision trees, then it became a high predictor. So probably getting a little convoluted there, but in other words, when you try to use your parameter sets, and again, this is a property of the resulting transients, and you try to use them to determine, well, if you're less than this or above this, are you going to give rise to a different outcome, right? If you form these decision trees, this is quite often a very important indicator, and we see that KMNE again. So this is kind of combining these different methods that I was talking about here, just significant testing, just doing what is statistically significantly different between the sets. These aren't ordered, these are all considered different between the sets, versus random forest, and these are ordering them as which are the best predictors. What we did was we kept everything pretty much on the same line here, so you can see that these were important to both. That's why, since order doesn't matter, there's some spaces on that one. We did it also when you looked at what actually fit the data, and you see that this KMNE is kind of there throughout, even with or without the data. But when we fit the data, that BR falls out. It doesn't become as much as important or indicative of anything. And this last column, this is ordered. These are the most significant parameters when we did that sensitivity with EFAST. And this is the one where you're just changing the parameter independent of the others. So just like we did in the previous model, we then went and looked at, well, this BR is important, this KMNE is important. Can we modulate that? And here we're looking at the original curve versus intervention at time 0. If you did it right away when you started the ventilation versus not until the end of ventilation or later. All right, and so we're seeing the different results, and quite often when the ventilator turns off, you still just go back to that where you were going to be anyways. But you see in these scenarios, if you actually gave BR, you could bring yourself back up to a resolved state, right? So you have the insult of the ventilator, right? Under normal conditions, you would end up at this persistent inflammation, but there are cases where we can actually pull it up to recovery. And same with the KMN here. And these are the associated macrophage transients for these outcomes with the different, right? So what's surprising is I think that the KME doesn't really change the M1s right? Even though when you change this, you're changing the amount of damage caused by the epithelial, but that's not causing a huge change in the feedback and therefore modulating the M1 response. So as I've mentioned throughout, and I hope we have a few minutes for questions here, our main thing is we want to be able to add in aging and kind of get more comprehensive data in all of these scenarios where we systemic and local, right? And when you start including these other compartments, can we use data that's collected from the blood maybe more often with less data localized to kind of make some decisions? So I'm gonna end here with my collaborator sign here. I work with a lot of people from biomedical engineering and internal medicine, and my collaborators in mathematics and our PhD program.
Thank you so much, Angela. That was interesting talk. We actually are past our end time. So maybe, is there like one quick question?
I got confused with the 5-minute late thing. That was my fault.
Well, maybe I will ask one question. So I actually very interested in, the effect of aging on the immune system. So out of curiosity, do you have a sex-specific model for the immune system? Given that, men and women are somewhat different as well in terms of the immune system.
So like in our current proposals and whatnot, we're actually getting data from mice of different genders because now that's required, right? But in the past, all of our data that actually, I mean, in the wound model, we included estrogen because, the hope was that we would actually have data from different genders, and we do think that it will be important, right? There actually are estrogen receptors on these cells, so the presence of it. It may not be as much as gender as much as just presence of estrogen or not. Postmenopausal females tend to heal slower than they did premenopause. And so, I mean, it's not enough to do a type of intervention, but you would expect that that would be there. So I think that, this next round of data, we're going to have to go back and adapt that, add in the estrogen modulation to be able to account for the differences in gender, not only age.
Great. Well, thank you, Angela.
And I apologize for that. I should have adjusted my timer here.
Okay. Yeah, when it's in person, I can stand up and I'll give you a hand.
Well, that's what I thought the beep was. I was like, wait, am I out of time?
I actually didn't hear anything at all. So
it must be on my computer. It must have been on my computer.
All right. Well, thank you. And
thank you so much.
Yeah. Hope to see you in person soon.
Yes. Bye.